Afternoon, everyone. It is my pleasure to welcome you all to today's section on a characterization and a DNA barcoding of polychaete worm. I am Dr. Ravi, Center for Ocean Research, Satyabama Institute of Science and Technology. Today, we will discuss the importance of Polychaete taxonomy, the basic principles of classification, morphological identification, and final how the DNA barcoding has uh, review valuation species identification. This is introduction. I'd like to talk about the taxonomy. What is the taxonomy? Taxonomy is the foundation of the biological science. Before studying any organism, we must first identify and classify correctly. The polychaetes are ecologically important marine worms that play major role in nutrient cycling, sediment mixing, fisheries, and aquaculture. However, the identification of polychaete species is often difficult because many species look like similar. Today, we will learn both traditional and molecular method of identification. So, what is taxonomy? We already know about that. The taxonomy is the science of naming and identification and classifying living organism. It allows scientists around the world to communicate using one universal language. Without taxonomy, biodiversity studies, conservation, ecological monitoring, and molecular research could not be possible. And he is the father of modern taxonomy. He introduced the binomenclature system. Every organism has two scientific names like genus and species. For example, Homo sapiens. This name system is still using in the world. This is Linnaeus classification system. So living organisms are arranged in hierarchical levels. The kingdom, phylum, class, order, family, genus, species. So this system helps us to understand evolutionary relationship among organisms. So importance of the correct identification means many organisms may appear different, but belongs to the same species. Likewise, some species may almost identical, but are genetically different. Like morphological characters alone sometimes create confusion. Therefore, the scientific combine external morphology, internal morphology, molecular morphology to correct identification of organism. So comparing organisms like simple characteristics are often insufficient. Scientists examine many taxonomic characters together than the relying on the future. Accurate identification require careful observation and comparison. So animals with elongated bodies are broadly grouped into yes. These are some of the examples. This first one is devil cat and the ghost cat and mountain lion. This all animals look like similar, but they're genetically different. The screaming cat and boomer, panther and fox. So this is very basic. It look like basically similar, but the genetically is different. So this is what we want to identify how it is different from the one animal to another animal. So this is another example, like what is difference between the animal, like horse and crocodile. So we can compare both the animal, like horse eyes, ears, and teeth, weight of the animal, location, livelihood of eating. So this kind of characteristics are majorly we found uh, from two animals. As we see this, there is very these two animals are having very broad differentiation, but we couldn't find similar species. And next one is the two word genus and species naming system for organism. Example, Homo sapien, how it generate it is by nomenclature. And another thing is this the like structure of the plant, animal, and fungi, protista, and everything. So this is basic classification of animal. Like the animals are divided into two parts: the vertebrate and invertebrate. So vertebrate, it comes under reptiles, fish, animal, and birds and mammals. For example, the invertebrate, like a protozoa, flatworm, like that. So we are in this study, we are going to see about the flatworms, roundworms, and segmented worms. So this, for example, the invertebrates, sponges, worms, and there are phyla of worms as three different phylum, like flatworms. Platforms are platforms, roundworms, and segmented worms. Among these segmented worms, Polychaete belongs to the animalia. The worm body characterization, or when the comparing when characterizing the worms, we observe bodies summative, like bilateral segmentation, protostotum, peristotum, and parapodia, kete, digestive systems, and reproductive organism. These features help distinguish different families and species. Next, flatworms have flatted bodies. Flatted bodies. We found out all over the world that more than 10,000 species platform bodies are available. So most are parasitic tapeworms, like it is mostly present in liver and livers. Some of the flatworms, like non-parasitic types, also available. For example, roundworms. Roundworms are cylindrical and mostly parasitics. They usually present in the human body to stay alive, feed, and reproduce, like a roundworm. Infection does not tend to cause any notable symptoms, so we need to identify very accurately. And this is segmented worms. And in the segmented worms only, we have different, like earthworm, polychaete worms, like that. So now we come into the polychaete worm characterization classification. Polychaete worms belongs to the kingdom Animaliae, phylum Animalia. Animal class Polychaeta. One common example is snails. The polychaete inhabit nearly all marine ecosystems. They are excellent bioindicator of marine environment. Diversity of polychaetes. Polychaetes are highly diverse. And ecological niches including sand, mud, coral reef, deep sea, hydrothermal vents, and mangroves. The diversity makes identification challenges. General anatomy of polychaete. Important structure includes the peristratum, antennae, eyes, pulps, parapodia, chetae, kiri, pydium. Each family possesses unique combination of these structures. These characters are essential for uh, taxonomic identification. The diversity of polychaete exhibit remarkable diversity in this picture. Uh, Firstly, we have to see the body size and the color of the organism, feeding habit where it is present in the ocean, habitat also we need to note it down, and how it is doing it involving the reproduction system strategy. So understanding this diversity uh, help us to study the marine biodiversity and ecosystem health. Now we come into the and one more thing. I want to add some point about the morphological, like it depends on the microscopic identification. Also, we need to take concern. So we need to confirm how many segments are present, and the shape of the parasite also we need to the parapodia also we need to confirm, and the type of kete, how it is diplo or dry, like that we need to check with microscopic identification, and we need to confirm the jaws, how the jaw present eyes, and a pair of proboscis, and some polychaete may have the gills also. These features are we need to record before going for the molecular identification. So now we come into the DNA barcoding. Yes. So uh, why we need to do DNA barcoding? The morphological alone cannot always distinguish closely related species. Uh, some species and juveniles forms often appear identically. So DNA barcoding provides an accurate and rapid method for species identification. It complements traditional taxonomy. And next, uh, yeah, this is the uh, clear picture of how to do the DNA barcoding. So first, whenever we isolate the polychaete ball, we need to confirm traditional identification. Then only we go to next level of identification. So once we collect the animal, we need to do the DNA extraction. The first step is collecting the healthy specimens, because some animal may cut during the collection of animal. It is very sensitive. It can easily cut the animal. So if we take the cutted animal or a cutted species, it may be the cross contamination. So that's why only we need to collect only the healthy animal and the full animal. We need to collect and wash it properly and store it. Storage is also very important. So once we did the storage, we need to do the DNA uh, extraction from the body tissue using commercial uh, DNA extraction kits. The quality of our DNA also we need to check before going for the PCR, because sometimes the DNA quantity we won't get because of the it is easily degradable animal. So sometimes it can easily degrade the animal and we won't get the much DNA. So we must confirm the DNA quantification before go for the PCR amplification. So and for PCR, the standard barcode gene for animal use mitochondrial cytochrome oxidase 1 gene. And sometimes this particular gene is work it happening because we need to use the different combination of primers. So, and sometimes it may amplify from 18S primers also. So we need to confirm with both the primers like CO1 and 18S also we need to confirm. And approximately the CO1 gene, approximately it's 720 base pair or amplified using PCR. And the amplified product are confirmed using agricultural electrophoresis. A clear DNA band indicate the positive or negative or how it is correct base pair or not. So this is what we want to confirm. And once the PCR product are purified and sent it for the sequencing, and the obtained sequence are carefully edited and assembled, and the low quality sequence are removed from the data. And the sequence analysis or compare with the database such as GenBank. Bold GenBank is very common common platform for submit the sequence, but the Bold system is very challenging one. So we need to give n number of data. So using and we usually the species similarity above 98 or 99% indicates the current identification. Below that, we won't consider the sequence. And next level is the phylogenetic analysis. After sequencing comparison, the phylogenetic trees are constructed. It uses a very common method like a neighbor joining method, and a maximum likelihood method also we use. And this trees reveal the evolutionary relationship among the species. And what is the DNA barcoding advantages means we can identify using fast identification, high accuracy, global standard, detection of cryptic species, and support for biodiversity conservation. Sometimes it is more important for the better ecological monitoring. Mostly this particular methodology we applied into the study areas like marine biodiversity, environmental monitoring, fisheries, aquaculture. We easily identify the species diversity also. So intensive species detection and Mostly we use the conservation biology and more importantly the uh, climate change studies also we use. And this is some of the picture I like to show in this slide. We usually whenever we go for the sampling, like that, we prepare everything in our boat. We have to take a tap sampler and the uh, stainer, rose Bengal blue stainers and formalin and ice pack also we used to take, and sometimes we will take the small centrifuge for DNA extraction in on spot. So we try all the method to produce a very good result from the polychaete worm. So once we collect the animal, we do the microscopic observation uh, um, with the external appearance. So how long the worm, like a dorsal view of the full worm, vertical heads and dorsal head, and tail regions also we need to confirm. Once we confirm those things, we couldn't say that this one is comes under we can say this maybe comes under the nearest species, but we cannot go, we cannot 100% confirm that it is comes under the nearest species level. So we need to confirm. We need to keys uh, anterior heterozono and the mid body of monopoly and posterior homologous like that. We need to key case identification and the jaws and everything we need to confirm. So whenever we confirm this morphological identification, it won't give the exact species identification results uh, because I already tell you that sometimes we will collect the samples the adult worm, sometimes juvenile worms. It won't be the similar all the time, so sometimes it makes false result also. So that's why only we use the uh, DNA sequencing method. And this is blast technique and the phylogenetic analysis and species identification for this. And this is actually the bold system for and look like. Okay, so whenever we need to submit our sequence, we need to log in properly with the exact values. And before submitting our sequence, we need to give the more information like you can see the here we need to give proper sample ID, and before we need to submit our sequence, we need to submit our sample to ZSI, and they will provide the museum ID. This ID we need to give here, otherwise we cannot submit the sequence details. So we need to get the museum ID also, and we need to give the information like where we collected the samples, latitude, longitude, and that exact name of the place and the district and state also we need to give, and we need to give the date of collection and collector name also, who are all the involved in this work. We need to give the names, and how many meters we collected, and how depth we found the animals also we need to check. And the sequence ID also we need to give, and in this sequence ID we need to give the what are the primers we use, and the sequence also we need to submit. Uh, they will rerun in their own in bioinformatics system, and each and every menu they will take at least 1 week for confirming. Once first we need to finish the identifier taxonomy, so it will take 1 week. Then we go for species identification. Then only we need to submit the collection data, and it is one by one procedure. Next. So once we finish our sequence data submission, they will provide like a DNA illustrative to be a DNA barcode structure. So this is the exact DNA barcode. This is called the DNA barcode, and nowadays they're providing another ID called like a QR code. Also, they are providing. So for instead of DNA barcode. So I like to show some of the picture for your knowledge. So this is a whole animal we need to give like this picture. So this is also very important. What kind of picture we are giving to the bowl system? So we need to upload the whole animal body system and the jaws and kidneys and the mouth part also we need to give, and the tail part, and how many segments are present. That's also we need to count and give the exact results. So anyway, we need to give the latitude, longitude. That information also it create the maps in that bolt system. So if you go there, the particular place, you will find the same animal. This is what bolt system say. And in our university, Center for Ocean Research, Satyabama Ocean Research Field Facility, we have the uh, exact uh, Polychaete REA system. So it is designed from our department. So we are maintaining here Parpaisa madrasi species. So we are continuously maintaining the three generation of polychaete worm in this culture system. So if you are anyone interested, you can visit the Satyabama Ocean Research Field Facility. It is already always open for students, so they can come and utilize the facility for the research work, or if they want to see the real marine worms, you can come here and visit our laboratory. Thank you. And another one more thing I want to share. Um, so many of them they need to think about why we want to study about the DNA for coding. And what is the future perspective of the study? Also, they are thinking, especially this is more important for classical morphological identification and molecular biology also. It involves a lot of study area also, like eDNA technology, metagenomics, artificial intelligence also. Nowadays, they are involving in this work. And this particularly, I need to give some point. The correct species identification is the backbone of the marine biology. Traditional morphological remains essential, but DNA barcoding provides greater accuracy, especially for cryptic species. By integrating both approaches, we can better understanding marine biodiversity support, conservation effects, and aquaculture research. And I like to share some more research on my work. So recently I had Bay of Bengal a intergovernmental research project. From that research project, we had a chance to visit University of Princhongla. So I like to share some of the clip on this project is very interesting. It's more related to the microplastic pollution, and it's co-contaminant with biofilm and other particles. So I like to share some.
Satyabama Institute of Science and Technology, Chennai, a Category 1 institution and NAAC A++ accredited university, leading innovation in science and engineering. At the forefront of marine and environmental research, Satyabama Center for Ocean Research explores the mysteries of our oceans. A partnership built on science, sustainability, and shared vision, connecting India and Thailand for a cleaner, healthier ocean. In collaboration with the Aquatic Science and Innovative Management Division of Prince of Songkla University, Hat Yai, Thailand, this partnership bridges two nations through science and sustainability. Through the Bay of Bengal Program Intergovernmental Organization, this initiative bridges nations for ocean sustainability. The joint, titled Studies on Microplastic Biofilm Formation and its Co-Contaminated Effects on the Marine Environment, explores how microplastics impact marine life and biodiversity. Led by Dr. M. Ravi and Dr. D. Indakandan from Satyabama, and Dr. Ekaran Rajaram and Dr. Korthaman Shwadaranku from Prince of Songkla University. Their research investigates how biofilms form on different microplastics and how toxic contaminants adhere and transfer to marine organisms. Through field and laboratory studies, the team analyzes contaminant accumulation, bioaccumulation, and toxicity in marine environments. Beyond research, this collaboration focuses on creating social awareness about the dangers of plastic pollution. Programs, workshops, and exchange initiatives, students and faculty engage in knowledge sharing and environmental education. The project empowers young researchers to protect our oceans and promote sustainable marine stewardship. We extend our sincere thanks to Dr. P. Krishnan, Director, BOBPIGO, for enabling this collaborative opportunity between India and Thailand. Together, we continue to advance marine science through innovation, collaboration, and commitment to a cleaner ocean.
And I thank you all for your kind attention. We will be happy to answer any questions.
Thank you, sir. Thank you, sir. Thank you for your insightful talk.